Okay, let's move on to updates from the cores. In my introduction this morning, I think I highlighted one of the changes in our organizational chart is that we are expanding at the urging of both NIH and the scientific review group that did our last competitive renewal, expanding the flow core into a bona fide immunology core, which means we're going to add, again, some immune assay services. So this is the chart I showed before. The flow core will remain intact as it is, but we're going to be adding multiple services on, including a QA/QC unit, ELISpot, an ADCC unit from Guido Ferrari, neutralization, HIV neutralization assay component, David Montefiore's lab, and then Georgia Tamaris' lab in binding assay. So we're going to have each PI or their designee come up and give you a brief overview of the types of assay services they will be offering, and give you an opportunity to ask some questions. So I think first up is Guido.
So considering that you guys are in your postprandial coma, I tried desperately to find a joke on immunology core on the website, and believe it or not, there is none. There are tons of immunology jokes that are probably written by the students, and they are absolutely not presentable in any site. I couldn't even believe what they were writing. So you get to live with the science. Sorry about that. So the core that, the portion of the cores that I'm supervising are two. One is related to cytotoxic response to HIV, whether they are mediated by cellular alone or cellular antibody components, and the ELISpot assay. And what that means is that we are trying to put this a little bit in perspective, because we are not just farming out assays. If you are looking a little bit at the way that this black dot, which is supposed to be the HIV virus, I was 5 years old when I drew this scheme here. But this black dot enters the cells and then replicates. At the very beginning, you can have the CTL killing. It's probably not very effective, but you have a mechanism like ADCC that can prevent this infection of the neutralizing antibody, and Celia will talk about that. And as the virus replicates and it makes himself known to the immune system, then the CTL start to play a role. And then again, at the time of virus budding, you can have antibody responses or CTL to prevent the release of infectious virus. This is why we are interested in these two components, eliminating cells that before they become infected or at the time they are infected. So the cytotoxic assay, there's a two-way. You can measure cytotoxicity at the level of the effector cells, and these are the degranulation using flow cytometry and the CD107 staining, or the cells that are releasing perforin. Or you can measure cytotoxicity at the level of the target cells. And this is what we are doing mainly right now. We are measuring granzyme B that is delivered into the target cells, or we are trying to quantify how many cells are eliminated by our responses. And I'm trying to keep my vocabulary very simple, because one of the only joke that I could have said is why we cannot understand immunologists. Well, because it's job security. And so I think this is the only thing I can say that I read. So this is the Grand Toxilux that we are applying for ADCC, but it cannot be applied to other, to study any other cytotoxic mechanism. What you are doing, you have effector cells and you have target cells. And you label your target cells in a way that you can identify them on your scatter plots. And you had the granzyme substrate And these permeate both cells, and when the effector can recognize the target, it will cleave this substrate and make the cells that are recognized fluorescent. So if this is your target cells and you define them as viable before you put in your assay, and you don't have effector, this is how they look. The granzyme positive cells would be in this quadrant. When you had this population, which is your effector cells, here is what happened. All 50% of the cells are recognized and become a granzyme B positive. The other methodology is the luck that we are using with SAD or HIV infected target. And in this way, we are, in this assay, we are trying to derive infected target cells by using either a swarm virus, which is just a field isolate grown in the cell line of your choice, or infectious molecular clones, which is the kind of the next generation of reagents that Christina Oxenbauer has made available to us. And these infectious molecular clones, what they have been engineered to have a luciferase gene reported inside. Once the HIV start to replicate, the luciferase is also activated, and that provide the luminescence signal to the assay. If you had your target, you can use this assay again with or without monoclonal antibodies or any kind of molecule that can recognize the infected cells and bridge between the target and the infector. Just is a 96 well plate, and this is how the well will look like. This is where the target alone are. They are very luminescent because infected. If you put the effector, you don't expect to see that much of difference if these effectors are not specific. But here, if we add, and the same if you had antibody from a seronegative or monoclonal antibody that are not of interest. If you had a monoclonal antibody that can recognize the HIV-infected cells or from a seropositive individual, you can see the level of luminescence go from 3,400 to 1,600. Applying this formula, you can calculate your percentage of killing, which is about 48% of cells are killed. This is the application that we're the assay are used to identify antibody-dependent cellular cytotoxicity responses that are either induced during the course of HIV infection or by vaccine candidates. And we would like to understand how they correlate with immune protection. And these are some of the project that we are collaborating, that we're using this assay for, the CHAVI-ID for generation monoclonal antibodies, so for CAVD, non-human primates study, HVTN, and PAVIG, these are NIH-sponsored study. We are also looking at the epitope specificity, breadth, synergy, and FC-related function of these monoclonal antibody, and see how we can combine them to be used in passive protection studies. Because the passive protection studies are usually done in non-human primates, we still are a little ignorant about how the NK functions is in the non-human primate. And it's definitely not the same as in human, and we don't have the same type of reagent, especially to characterize the Fc gamma receptor function in non-human primate that we have in humans. So right now we are just screening the animals before they are going to be utilized for this passive protection study to make sure that their NK functions are somehow similar. The other thing that we are doing is we are studying T-cell effector function against the virus-infected cells. And here we have some collaboration, not just with CHAVI-ID, but also with David Margolis and in the CFAR with Dr. Chen, who are trying to eliminate the pool of latent infected cells. And in order to do that, we are evaluating also the ability of some compounds that are called DART, and it will take another 10 minutes to go through it. But they are essentially capable of recognize the infected cells and recruit T cells that have a cytotoxic potential but are not necessarily antigen specific. So this is another project that we started. The ELISpot, probably a lot of you have read and are familiar. It's a very simple method to quantify the antigen-specific cells by using a 96-well membrane. In case of the interferon gamma, you use one monoclonal antibody, you're coating your wells, then you add antigens and cells. You let them sit overnight in order for the antigen to stimulate the cells that are antigen-specific. You remove the cells, and some of the interferon gamma is bound to this capturing antibody in orange. Then you add what we call the detecting antibody that it can be labeled to biotin or directly to an enzyme. It will form a sandwich that is insoluble, and it will precipitate upon the addition of the substrate. And each one of these spots that you can see easily from there, I hope, represent a cell that was able to recognize the antigen. And what this has been used in the past is to determine the magnitude of the antigen-specific cells that are circulating in the blood or in any sample that you like. You can map the epitopes that are recognized by these cells if you use the right antigens. And you're also able to determine which one is the subset of interest if you do some selection of the cells. This is currently performed under GCLP, and so is a sum of the ADCC assay. And if this is not in a big investigator-initiated collaboration yet, but we have a few things that may turn out to be useful in the next year or so. So this is really the people who are doing the work, and if you go on the CFAR website, there should be, other than my name, at least 3 person of a contact. And Joy is for the cytotoxic assay. Mark is for the ELISpot. He's on vacation as per noon, so he's not here. And Justin is for the ADCC, and all the others are always happy to collaborate with any other person at Duke. These are all the collaborators, and that's it.
Thank you, Guido.